Okay, so we say that a fat acid will contain a hydrocarbon chain which ends in a carboxyl functional group, and that fat acids are amphipathic. They have both hydrophilic and hydrophobic ends. Then I said that they can be divided into saturated and unsaturated fat acids. We have said that saturated fat acids have the maximum number of hydrogen atoms, the maximum possible number of hydrogen atoms. So saturated fat acids are saturated with the possible number of hydrogen atoms. And we gave the example of palmitic acid as an example of a saturated fat acid. Then we are saying that fats which are high in saturated fat acids, for example, animal fat, tend to be solid at room temperature. They tend to be solid at a room temperature. So the reason for this is due to the presence of van der Waals interaction that can exist between adjacent molecules. Remember that even if a molecule is electrically neutral, find that non-polar molecules can actually develop transient regions of weak positive charge and weak negative charge. So even if the molecule is electrically neutral, when we look at the non-polar molecules, these can develop transient or temporary weak positive charges and weak negative charges. So this comes about because of the constant motion of electrons. So that constant motion of electrons will cause some certain regions to have excess electrons. So that will be just a transit or a temporary excess of electrons. And then if other regions have excess electrons, it means that the other regions may have a temporary electron deficit. So they're going to have an electron deficit. So then having these opposite charges results into the van der Waals interaction that occurs between these molecules. So remember, even though van der Waals interactions or attractions are actually weak attractions, they are made strong when men occur among long hydrocarbon chains. So they become strong if many of them occur along a hydrocarbon chain. So these van der Waals interactions or attractions are the ones that tend to make a substance more solid because of the attraction, it prevents or limits the motion of its molecules. So no wonder we are saying that the fats that are high in saturated fat acids tend to be solid at room temperature because of the presence of the van der Waals interaction that exists between the adjacent molecules, which means that it limits the fluidity or the motion rather of the molecules. So fat acids high in saturated fat acids tend to be solid at room temperature. When you look at unsaturated fat acids, so if a fat acid has a double bond between one or more pairs of carbon atoms, it is said to be unsaturated. So if a fat acid has one double bond, one double carbon to carbon double bond, these are called or known as monounsaturated. If there are more than one, we are going to call them as polyunsaturated. So poly for men, mono for one. So remember we talked about the cis and trans configuration. And also when we talked about the isomerism, the cis and trans configuration. So when we look at unsaturated fats, we find that most of the fat acid, unsaturated fat acid, occur in a cis configuration. Remember I said that cis configuration means that the carbon chain is on the same side before and also after the double bond. That's what cis means. And then trans means that they are on the opposite side. So cis, same side. Trans means opposite sides. So this is an example of a trans double bond. This is cis double bond. So we can see the hydrogen atoms are on the same side. But when we look at the trans double bond, we find that these hydrogen atoms are on the opposite side. So we have this hydrogen atom up there, and we have this hydrogen atom down here. Cis double bond, all the hydrogen atoms are down here. So this is cis double bond, this is trans double bond. So most of the natural unsaturated fat acids will have a double bond with a cis configuration. So they're going to have this configuration. So we find that unsaturated fat acids actually are not fully saturated, just like the word unsaturated means. So they are not fully saturated with hydrogen. So they do not contain the maximum possible number of hydrogen atoms. So they are unsaturated. So I find that most of the fat acids, or most of the fats that contain a high proportion of saturated fat acids, they are going to tend to be liquid at room temperature. Remember I said that for the saturated fat acid, most of them tend to be solid at room temperature. And the reason for the unsaturated fat acids to be liquid at room temperature also has to do with the presence of the limitation provided for the existence of the van der Waals interaction. Remember, due to the van der Waals interactions in saturated fat acid, we find that they become solid. So we find that in an unsaturated fat acid, that double bond in the hydrocarbon prevents the fat acid from aligning closely with an adjacent chain. So in saturated fat acid, you're going to find that these adjacent chains are going to align closely, and henceforth that van der Waals interaction is able to take place. But in unsaturated ones, due to that double bond limits van der Waals interaction, and henceforth we find that this permits a fat acid to be in. Okay, so no wonder we find that these unsaturated fat acids tend to be liquid at room temperature, So additionally, we are saying that having that double bond will change the behavior of a molecule. Because remember what we talked about when we in the macromolecules class, free rotation cannot occur about a double carbon to carbon double bond as it can occur in a single bond. So when we look at this, it is actually going to mainly affect the melting point. It's going to affect whether a fat acid is going to be a solid or a liquid at room temperature. So when you look at a fat acid that has a lot of, or when you look at a fat that has a lot of unsaturated fat acid, it's going to have a low melting point because of the presence of the double bond, which causes a bend, and henceforth preventing the fat molecules from aligning closely with one another. And henceforth, this prevents the occurrence of strong or many van der Waals interactions, just as we have explained. So find that polyunsaturated fats with polyunsaturated fat acids are going to have low melting points due to that fact. So most saturated fats, as we mentioned, such as animal fats or butter, are solid at room temperature. They are solid at room temperature. So when you look at hydrogenation, so this is an industrial process which manufacturers can use. So they do hydrogenate or partially hydrogenate cooking oil. So for example, to make margarine or other foodstuffs. So during hydrogenation, addition of hydrogen atoms, we find that these unsaturated fat acids are converted to saturated fat acids. And henceforth, we find that these are made to be more solid at room temperature. So in the industry, the conversion of unsaturated fat acid to saturated fat acids can be done. So now what this leads is that this makes these fat acids to be less healthful. So they are not more healthy because they actually increase the risk of cardiovascular diseases. So they increase the risk of having cardiovascular diseases. So I find that someone can start having heart problems because of the fat deposit. And then, so I find that fat deposits can be made in the blood vessels. They can be made in the blood vessels, which is why it becomes a risk of cardiovascular diseases. So someone can have issues or diseases in their cardiovascular system. So the heart can be affected. So find that someone can actually develop hypertension, high blood pressure due to these fat acids. So no wonder they are not more helpful. And then also hydrogenation. We find that when these fat acids are actually artificially hydrogenated, the double bond can actually become rearranged, which can result in a trans configuration. Remember, I looked at the cis and the trans configuration. So when you look at the trans configuration, fat acids are actually unsaturated, but they tend to mimic the properties of saturated fat acids. So remember, when we look at the trans fat acids, they actually do not produce a bend or a flanking at the side of that double bond. So trans fat acids also tend to be solid also at room temperature. And henceforth, they also increase the risk of cardiovascular diseases. So during hydrogenation, we find that the double bonds can become rearranged to produce trans fat acids, which also increase risk of having cardiovascular diseases. And then remember the saturated fat acids also increase the risk of cardiovascular disease as well. And then we have glycerol. Remember I said that fat or lipids, lipids is esters of fat acids and alcohols. And I said that the triglycerides are going to contain glycerol and also fat acid chains. So when you look at glycerol, this is the molecule, this is the structure of glycerol. This is the structure of glycerol. So its molecular formula is C3, so it's going to be C, there are 3 C's, so 1, 2, 3. So C3, H8, so there are 8 hydrogen atoms, 8 hydrogen atoms. So C3H8O3, then there are 3 oxygens. So this is a molecular formula for glycerol. So C3H8O3. So when you look at glycerol, it's a simple 3 carbon, as we have explained, simple 3 carbon polyalcohol because of these OH groups, the OH group, which is going to combine with fat acid to synthesize triglyceride or N-glyceride lipid. So it says it's actually a colorless, viscous, and sweet tasting compound. So the presence of these hydroxyl groups, these 3 hydroxyl groups, is going to make this glycerol molecule to be an adsorbent and also to be water soluble. So glycerol is water soluble due to the presence of the hydroxyl groups. So remember, one of the properties of hydroxyl groups is that it is a polar. It is polar. It's a polar polar functional group. So as we mentioned, its chemical formula is C3H8O3. C3H8O3. Glycerol can be used as a solvent, as a sweetener, as a preservative, as an additive. So it can be added in food, can be added in pharmaceutical products, and also cosmetic products. Glycerol. Sure, we are familiar with glycerin. So glycerol can be used in cosmetic products, pharmaceutical products, drugs. In drugs, can be used in food, can be used as a preservative or as a solvent or as a sweetener. So it is produced from a reaction that is called saponification. So it's simply a reaction between sodium and a glyceride. So it's produced from lipids through saponification. So this is a process of saponification. So simply a fat plus sodium hydroxide. Through this process of saponification, we are going to produce glycerol, C3H8O3 glycerol molecule. So it's produced through the process of saponification. And then we have the triglycerides or the glycerides. So these are triglycerides, actually the most abundant lipids in living beings or in living organisms. So they are commonly called or known as fats. So these are very important form of reservation of energy. So they are good storage materials or molecules. Because we find that, as we explained on the functions of lipids, we find that when these lipids or triglycerides are metabolized, they actually yield more energy than can be obtained from carbohydrates. Furthermore, we also take note that carbohydrates, as we explained in the last classes, said carbohydrates actually can be transformed by enzymes into fats. In addition to that, also proteins actually can also be converted into fats. So we have what are called adipose. We have the adipose tissue, which is simply composed of fat. It's a fat tissue that is found in animals. And then this conversion can also happen in fruits and also in seeds of plants. So they are very important storage molecules. So a triglyceride consists of a glycerol that is joined to 3 fat acids. So it has glycerol connected to 3 fat acids. No wonder it's called tri for 3. consists of a glycerol having 3 fat acids. So we've already looked at glycerol, it being a 3-carbon alcohol, and it has 3 hydroxyl groups, while a fat acid has a carboxyl group at one end and an unbranched long hydrocarbon chain. So remember we looked at condensation reactions when we looked at the macromolecules. And we said that these condensation reactions are going to link monomers to form macromolecules. So the condensation reactions or the dehydration reactions are involved in the formation of triglycerides. So for it to be tri, it means that 3 reactions need to take place. So 3 reactions need to take place. So remember, in each reaction of dehydration, in each dehydration reaction, find that an equivalent of a water molecule is removed as one of the glycerol hydroxyl groups reacts with the carboxyl group of the fat acid. So this is the carboxyl group, COOH. So one of the hydroxyl groups of the glycerol is going to react with the hydroxyl group of a fat acid, which is going to result in a link or a linkage or a bond, which is called an ester linkage. So what is going to form is an ester linkage. So since it takes place 3 times, it has to happen 3 times for us to have a triglyceride. When there's a first reaction, it means that this reaction, what will be formed is a monoglyceride. Mono, since it's just 1, so it's going to be mono. When it is 2, or rather the second reaction, which means that the second fat acid has been joined to the glycerol, this is going to be a diglyceride. And then when the third fat acid has now joined with the glycerol molecule, it now becomes a triglyceride. So remember, the hydroxyl group of glycerol will react with the hydroxyl group of the carboxylic acid group or the carboxyl functional group of the fat acid to form an ester linkage. So this is the process of esterification or the formation of the triglyceride. So we have the glycerol molecule, then we have the fat acid, glycerol molecule and a fat acid. So for example, we have these are 3 fat acids. This fat acid, that fat acid, and that fat acid. These reacting, remember it's a 3-step reaction, or the condensation reaction takes place 3 times or happens 3 times. So it means that 3 molecules of water are going to be released, 1 in each reaction to give the triglyceride. So the OH of this glycerol will react with the OH of the carboxyl group of this fat acid to produce the ester linkage. Similarly, this one also react and also this one to form the ester linkage and henceforth forming a triglyceride. So this is the process. So this is the ester linkage, one in gray. So it's depicting an ester linkage. So during the formation of these glycerides, so you find that usual actually there are different fat acids that react with the glycerol. So maybe different fat acids. So remember from what we've looked at, what type of fat acid is this? Saturated or unsaturated?
Saturated.
This is a saturated fat acid, isn't it? And then this one?
That's an unsaturated.
Unsaturated, and then what type?
Monounsaturated.
And yes, go further.
Monounsaturated.
It's a cis, isn't it? It's also a cis, right?
Yes.
It has a cis configuration. Yes. Was the hydrogen bond, the hydrogen atoms are on the same side. This one similar is the what? Unsaturated, isn't it?
Yes, unsaturated.
It has two double bonds, two carbon to carbon double bonds. And then it also has a cis configuration. It has a cis configuration. So at that double bond, double bond there, it's going to form a kink or a bend. So no wonder this picture is showing this kink or a bend. It's going to form that bend. Remember when we talked about the unsaturated and the saturated, and why unsaturated are liquid and why saturated are solid at room temperature. So it forms a kink. So this one is also going to form a kink wherever there is a carbon to carbon double bond. Then palmitic acid is a straight chain, or it's a saturated fat acid, so it has a straight chain. Then when you look at oleic acid is a monounsaturated It's a monounsaturated fat acid. Then we have linoleic acid, which is a polyunsaturated. Remember, poly having more than one double bond. So it also forms a bend or a kink there. So we see this triglyceride molecule or triacylglycerol molecule. So it contains 3 fat acids. We have palmitic acid, which is a saturated, and we also have these unsaturated. So they are different. So we have different fat acids joined to the glycerol molecule. So during the digestion, this triglyceride can actually be hydrolyzed. So through the process of hydrolysis, these triacylglycerols are able to be hydrolyzed to produce the monomer units. So we're going to have the glycerol and also the fat acids that joined with the glycerol. So we're going to have the monomers being produced through the process of hydrolysis. So when you look at the diacylglycerol, or the diglyceride, it's a very important component involved for sending signals. So cellular signaling, as we are going to look at it further, that diacylglycerol is a very important molecule for cellular signals within the cell.